The second process in oxidative phosphorylation is chemiosmosis. Chemiosmosis refers to the movement of proton from high concentration to low concentration through ATP synthase. ATP synthase catalyzes the synthesis of ATP from an ADP and phosphate. The difference in the concentration of proton between the matrix and the intermembrane space was previously established through electron transport chain. Because of this proton gradient, proton will diffuse from the intermembrane space into matrix via ATP synthase. The flow of proton will releases energy, which is used by the ATP synthase to phosphorylate ADP into ATP. This process will continue as long as there is difference in the concentration of proton across the inner membrane of mitochondrion. Next, we will discuss the products of oxidative phosphorylation. First, oxidation of one NADH in the electron transport chain will be able to produce enough proton motif force to synthesize 3 ATP during chemiosmosis. Secondly, for each FADH2 oxidation in electron transport chain, the proton motif force produced can be used to synthesize only 2 ATP by chemiosmosis. Since glycolysis will produce 2 NADH, this means that a total of 6 ATP can be produced during oxidative phosphorylation. Link reaction also produce 2 NADH, which also means 6 ATP is produced via oxidative phosphorylation. Krebs cycles, there are 6 total NADH produced, which is equivalent to 18 ATP. In addition to NADH, Krebs cycle also produce 2 FADH2 molecules, which is equivalent to 4 ATP. For Krebs cycle alone, a total of 22 ATP is produced via oxidative phosphorylation. By counting all the numbers of ATP produced during oxidative phosphorylation, for each glucose oxidized, total of 34 ATPs can be produced. Don't forget the ATP molecules produced via substrate level phosphorylation, which is net of 2 ATP during glycolysis and net of 2 ATP during Krebs cycle, which will add up to the 38 total ATP produced for each glucose molecule oxidized.